be done on earth as it is in heaven. Give us this day our daily bread, and forgive us our trespasses, as we forgive those who trespass against us. And lead us not into temptation, but deliver us from the evil one. In Christ Jesus our Lord, for thine is the kingdom, the power, the glory forever and ever. Amen. And now you are with Daniel to give you the introduction about the day.
So the day's divided into 3 talks. Originally, we were going to start with theology, then history, then finish off with a bonus talk on evolution and Christian faith. But we're doing it in reverse order. So we're going to start with a bonus talk because he had something to attend to today, but he still wanted to give the talk, so he's joining us live. Can you all hear me? Okay. So we're going to start with the first talk, evolution and DNA. Is that right, Abuna?
It's basically faith and reason, and will apply to the theory of evolution.
So science, the faith, theories, because this is what usually people are attacked with first. And Abuna is going to show us how that's not the case. Then the second talk will be the science, because that shows us the theology, how they approach the world, approach science. And us as Orthodox, this is extremely important. Already there, but we don't look for it. So that will be the second talk given by. And the third talk will be the history of Christianity's interaction with science. Because not only are people attacked from the scientific point of view, in conflict from the beginning of against.
Hello?
Sorry. The 3 talks will cover those different aspects. So they all address points of this debate, the faith and. So we'll start off with Abuna today, and after he's done, he will take questions. If you ask questions, you need to stand right up here and look at the camera so he can see you. Okay? And if you in the front 6 rows as well, he can see you now. So everyone wave at him. Okay.
Pray for me.
And in your books, your little booklets, there are questions to go with his talk. So that way, if you want to take notes under those questions, those topics, you can do so. All right. So with all that said, please, you may begin.
Okay. Absolve me, Abi. Can you all hear me well first? Because as Daniel is speaking, it sometimes clips the sound. It doesn't matter for me on this side as long as you can hear me well on your side. So can everybody hear me well?
Yeah.
Yes, okay. In the name of the Father, and the Son, and the Holy Spirit, one God, to whom is the glory forever and ever. Amen. Thank you very much. I would have loved to come. I love the fathers there very much, and the church, and all of the servants there. So it was really a conflict on my behalf, on my part, that I was stuck in 2 things and I couldn't do anything. So I had to do it remotely because I wanted to be with you. Still do the talk and listen to your questions. And if you cannot fit everything today, we can have it through email or whatever you like. Orthodox apologetics. Well, before I delve into the subject of the evolution, I just want to tell you where it is on my map when I do apologetics in order to see where we're coming from. Because in Acts, we're doing a comprehensive approach to it, and I do this in the class that is offered every spring. It's called Apologetics 1 in Acts. It's about 16 lectures long, 3 hours every time. So you can tell it. We cannot cover everything, obviously, here. Just I wanted to know where the evolution discussion is part of a big umbrella I have developed over the years in order to develop a pattern for Orthodox apologetics, because it's very unique compared to any other apologetics. It has to be answering even things about the church. So it all comes under the umbrella of be ready Revere Christ in your heart. Be ready to give an answer to anyone who asks you about the reason of the hope, for the hope that's in you, and do this in meekness and reverence. 1 Peter 3:15. So in a excuse me, in 5 minutes, I just want to give you the map of apologetics that I do in the class, and this you'll see why where evolution fits in, and I will touch again on it when we do the talk. So I divide it into 4 levels. Every level feeds into the other. The first level is the basic and the most difficult one, which is God's existence, or the man Jesus existed. If he's existed, is he God? Is the resurrection true? So there's 2 aspects of it. Is there a designer? Or is there God? Or is there someone behind this world? And if this man Jesus claiming that he's God, is that true? Is there any evidence for this? And definitely, all of this level does not touch the Bible, and the Bible is not used in it or any religious terms, because it will be completely not part of the architecture I'm building, because anybody who's coming from outside wants to have an answer, or from inside, and we use the Bible, he was already turned off because it's an inside answer. So we have to have all the answers from outside the Bible. So let me give you an idea. Let me first shrink it in order for us to know what I cover in it. Philosophy and logic. So we're going to be in science today, but history is usable because it's history outside the Bible. Philosophy is usable. It's the philosophical approaches about the value and the purpose and the meaning of life. And logic, and there is some very crystal approach I used here, really came up with it over the years as well, that logically can prove so many things about the authenticity of this new movement called Christianity. But it's not our talk today. So our talk today is in the realm of science. So science, history, philosophy, logic, without using the Bible, or without pre-notion about the existence of God, is what gets covered in level 1. And as you can tell, that's the harder one. It takes about 8 lectures out of the 16 to give enough depth in this, yet I not cover everything, but enough for the student to be really feeling comfortable on how to research next. And you're all maybe college students. The idea of going to college is not to graduate. If you're an engineer or doctor, you're not ready to perform, but you're given a license to learn more about the field. That's really what your graduation is. That if a body of your field recognizes you, that you're now at the level to start learning in your field, you're authorized to do it. This is really what your degree is. So this is what I do here as well, is that enable the students to know how to search, which direction to search in more. But with enough depth, but more open questions for them to go and do research on. So today, we're in the science area. So the science coming, we're going to talk about faith and science, or faith and the interface between faith and science. Is it existing? Is it non-existing? The 2 most important topics in science, evolution and cosmology, because the first one says that there is a certain mechanism that accounts for the origin of life. It's called evolution. And the second one claims that the world came by itself, on its own, a world out of nothing, as Lawrence Krauss claimed it. And that's a series of other talks. With it, I conclude scientifically that very clearly the world has a beginning. And this has a lot of quantum mechanics, Einstein's equations, Schrödinger equations, the history of cosmology, the schism that happened between the church and science based on cosmology, Galileo, and all of this history has to be covered in order to see how we reach today. But these 2 are the most fierce ones. And here is my claim. I want you to write this down. If evolution cannot account for the origin of life, and if cosmology cannot prove that the world started by itself, then there must be one outside of life and outside of material that had started life and the universe. Again, if and I underline cannot here, because cannot means scientifically, we'll talk today that science has a lot of holes when it claims that evolution is the explanation for the origin of life. So let me repeat it again. This is the logic. If evolution cannot account for the origin of life, and if cosmological facts today cannot prove that the world started by itself, therefore, there must be a cause outside of life and outside of material that had started life and started material. So the word cannot here is the key. So what we're embarking on today is to prove with enough teeth. I have actually 7 claims of challenges to evolution as it is today. So the word cannot means I will present scientific evidence and claims and hypotheses that challenges the claim that evolution explains the origin of life. Because I am saying no, it cannot. And it cannot because the Bible says God exists. No, it cannot because scientifically the leaps that evolutionists make are not scientific. And they will go through this here. And there are 7 fields of engineering that help in this: mechanical design, control systems, systems and signals, rule of thermodynamics, energy conversion, electrical engineering, computer engineering and architecture of digital systems, machine learning and artificial intelligence, information theory and digital communication theory, all exist in biology. And these fields are not taught as under biology discipline or biochemistry, but they all exist. And I don't have enough time to show you the different systems. I will see some of them today, and if you want to have a follow-up, but this is the discipline in engineering that is needed now in understanding how biology works. And when biologists don't find an explanation because they are not disciplined in this system, they claim, they jump, and they say evolution found a way, or evolution, this must be attributed to an ancestor, and all of these are completely unfounded. I'm going to show you examples from biological systems today. So let's get into the talk. So remember again, in level 1, if evolution cannot account for the origin of life, and cosmology, today cosmological proofs cannot account that the world started by itself, in fact, it's the opposite, but that's not the topic today, then there must be a cause outside both of them that started life and started the universe. So level 1 covers this. History and philosophy and logic are all approaches also to the existence of God, and of course it cannot be from the Bible because it would be self-defined. If we prove that God exists, which religion does he exist in? And then I do a comparison with other religions, but I focus in the class on Mormonism because of it's a very American faith, and also for the lack of time of focusing on the other. If Christianity is the faith, which Christianity is it? Is it apostolic or Protestant? So this is a very, very important topic for us within the church, understanding the creation, understanding the fall, understanding the communion, how it is related to fixing the fall, answering, do this in remembrance of me, and faith and works in Galatians and Romans. And then when we find it's the apostolic Christianity, then we dig deeper into our faith with several topics. There is a reason I choose those, but it's not important for us today. So that's basically the umbrella of Orthodox apologetics, because when you go to any Protestant school and they give you apologetics, it's usually biblical apologetics, and it doesn't go at all into this, especially when you come into the communion. So I want to give a comprehensive mindset or a way that you can do research further by knowing that you can layer the arguments or layer where the listener is or layer where your topic is, which level you're in. So we are in level 1, which is the hardest one today, talking about challenges to the theory of evolution. So let's start, and hope Abuna prays for us that it goes smoothly and we all benefit from the presence of the Holy Spirit who teaches us all. Theory of evolution, a scientific challenge to its overreach. So evolution definitely exists. It's a brilliant mechanism. I'm not going to say that God put it, because we're not talking about we're not saying God exists. But we're saying here that this mechanism is brilliant, that there is an adaptation, there is the species can acclimate themselves, modify themselves, progress, and on its own. There is no interference that's needed. And this is the sign of great, actually, intelligence, that once a species is there, it modifies itself to adapt to what's around it. So evolution, in that sense, is very, very brilliant. When does it overreach? When they claim that it is behind the origin of life. And this is where we're going to put some challenges today. Faith and science, is there any interface between them? Faith and science, is there any interaction between them? Interface is a term used when two different entities interact with one another. For example, when you put your USB into the computer, there is a USB interface between the computer and the USB drive. And in fact, for the computer to talk to the USB, there's something called device driver that creates a language between the two. Or a GUI, a graphical user interface. It's when a human interacts with a machine, a computer, for example, the screen, or input-output devices more accurately, or the keyboard, how a computer enters something a human enters something in a computer, and how the computer displays something to the human. All of these are interfaces. Is there an interface between faith and science? So there are two different circles. The former is based on belief, but supported by initial eyewitnesses, especially the faith in the resurrection, while the latter is empirical, is about testing and testability, which we'll see a lot of its non-existence when we see the claims that evolution does in jumping in order to cover gaps in their arguments. But when science here is when science starts poking into the faith. When science makes a metaphysical conclusion, then it crossed its boundaries and entered into the boundary of philosophy, which is a faith arena. Philosophy is a truth claim about a state of things. Science is about empirical data. So when science uses empirical data to make a philosophical conclusion, that's a faith statement. So since evolution accounts for the origin of life, therefore there is no God. This therefore, assuming that the science is correct, which is what we're challenging today, this therefore is a philosophical claim. It's a metaphysical, we call it a second order assertion, a conclusion. Science is not, this is actually, by the way, is a field called the philosophy of science. Philosophy of science can do conclusions. Science does empirical data and can do hypothesis, and then can do experimentation and then do conclusions. So all of this circle is within science. When it does a conclusion, therefore there is no God, now the science has done a metaphysical conclusion. Then the faith has to answer. But the faith is in a different circle. So the faith, which is what I'm trying to do today, I'm not answering from faith, I'm answering in the field of science, because science made a claim against my faith. So I'm going to say, I am in that faith, but I'm not going to answer you from the Bible or from my faith. I'm going to answer you from your claims that your assertions, your conclusions, and your science has holes in it, scientific holes in it. So a philosophical answer may suit those who are in the faith already, that's definitely, but that's not the topic for today. We're assuming we want to answer those outside the faith or poke holes in their claims. But the same scientific answer is extremely needed when those of the empirical community, with those of the empirical community. In other words, if a scientist attacks the Bible by claiming that, here is the two claims always, evolution accounts for the origin of life and that the world has no beginning, or eternal in the past, or a bouncing universe, all of these models answer them, or a multiverse, or it's just not the topic of today, but these are my two biggest fields of delving in order to, as we said at the beginning, to prove that these claims cannot stand on science. They have holes in them. In fact, we answered, especially with cosmology, that the world has a beginning scientifically, and we know exactly when that beginning is. Then the scientist has used his data, we know exactly scientifically when that beginning is, not biblically. Then the scientist has used his data to make a conclusion or a philosophical statement. The philosophical statement is the absence of God, the absence of a creator who can give life, the absence of a creator that can create material out of nothing. then we can challenge this conclusion as the men of faith. But we can challenge it not from the Bible. I avoid that 100%, because if I use the Bible, say, here is a scientific fact here. If anything else is different from what I claimed, I will shatter the Bible. And that's why, on purpose, my apologetics approach is not biblical apologetics, but rather facing scientists in their science. Then I'm safe. And the Bible has its own field. Things in it scientifically are correct, but maybe not precise. That's different topic. But definitely, you'll not find anything I'm going to use in the Bible today. The conclusion, the absence of God today, became in the scientific community as a given. So these arrows basically we're answering back from science. So we're attacked on the faith by them saying there is no God, and we're answering back from their own science that your claims are lacking validity. Let's remember again, if evolution cannot account for the origin of life, and if cosmological research cannot account for the origin of the universe, then there must be a creator. And the word cannot here means that the claims they offer scientifically are extremely challengeable and extremely full of gaps. That's what we're about. I'm repeating it in order to remember that concept, and I was able to crystallize it very recently to deliver it in very short statement. But today, people move with scientific dictatorship. There is an absence of God. Evolution is the origin of life by default. You have to align all of your thinking. If you find any scientific discovery, you're not allowed to have any other conclusion. Make any claims, make perhaps, make probably, whatever it may be, but you have to stick to the theory of evolution. So that actually is scientific dictatorship, and you're all exposed to it by professors who don't have multidisciplinary field in order to see what they say. If it's listened to by another field, it would be laughable. It will not stand ground at all. The conclusion, the absence of God today became a fact, not a claim, not a hypothesis in the scientific community. As a reaction to this, people of the faith come to the stage to defend. But the defense is not to answer science from the Bible, because the Bible is not a scientific book, but rather to challenge the conclusions the scientists draw from their data. That challenge has to be scientific. By offering other plausible data, by offering equally plausible conclusions from the data, and or by showing that the conclusions are at a large gap from the data. And that is the scientific method or methodology that you're all familiar with. Observation, hypothesis, experimentation, conclusion. Unfortunately, conclusions sometimes are drawn already. You just have to put your scientific papers in order to publish, in order to get grants. Actually, there's publication journals that when people publish something related to the presence of a creator, very scientific approach, these editors were threatened to shut their magazines down. I want you to watch actually Darwin's Dilemma, a YouTube video, very, very, very useful. So the challenge has to be scientific by one or the three methods: offering other plausible data besides the data that was offered, offering equally plausible conclusion from the data that was found, and by showing that the conclusions that are made by them, by evolutionists, by them means that evolution that claim that evolution is the answer to the origin of life. When I say this, evolution again exists, it has to exist, it is a very intelligent system. It doesn't have foresight, it doesn't know where it's going, but it's the presence of a very intelligent mechanism that species can adapt based on what they are surrounded with. So here is what I said. Does God exist? Evolution theory claims explaining the origin of life. Cosmology claims Big Bang is the beginning of the universe by itself, on its own. Therefore, there is no intelligent creator. That's basically what we're in day in and day out. If we prove scientifically that evolution theory cannot account with scientific rigor for the origin of life, and cosmology cannot prove that the universe started by itself, then there is an intelligent creator. We call him God. The word cannot here means the scientific evidence that evolution theory proposes is having major challenges to the point that the theory is not able to explain the origin of life. Second, the cosmological theories are having major scientific challenges to the claim that the world, I mean here the universe, cannot come by itself. And there is, again, this is a separate topic, but there is ample, very, very beautiful descriptions of what was there at the beginning and what did the Hubble telescope prove. And all of these things are, as I said, everything has to be scientifically sound. Evolution undeniably exists, but we shall portray borders for it. Science presented as borderless, and this is a type of faith, in fact. You start from its existence and then you fit the data to it. You believe in evolution first, then do science. So there are several of them, but related to the previous conference we did with lovely servants, Michael and Mary and Kevin, and you get the blessings of listening to Michael's talk today as well. So we're here putting just borders between its claims. It is existing, definitely, but there are certain challenges that makes its borders to be not overreaching that to explain the origin of life. I'll focus today on the information enigma, and I will touch also on probability and chance a little bit. This is for the conference. Also, by the way, for this as well, I would love that all of us share. Thank God that Daniel is doing this as well. The goal of this conference is we need your feedback, share ideas on annual basis, encourage youth to publish, hoping that you can publish papers, even challenging to what I'm saying today on the blog. I was trying to make Coptic Apologetics as this actually website has the talks of the previous conference. Go to see the fathers, Abuna, my weak self, and whenever we have another conference, to see the bishops. Get to know Acts. It's a very, very strong educational system that we're building up in the diocese with very, very pure education. And Abuna Daniel has a huge lion's share in courses that he gives, and I just had a course with him for the fathers that really was very, very uplifting. We made a Facebook page, but it's not maintained well. I want to focus on one entity, which is published on Coptic Apologetics. Origin of life is exactly the origin of information. Here's the big question: where did information come from? And information, as you will see, has certain different forms, but it's the sign of an intelligent being. So life actually equals information, not chemistry, and we'll see how. Even with some things in the talk, how my claim will stand. Examples of information: a musical note is an information. It's a digital information. Every note is standing by itself. A Word document is digital information. Every letter is standing by itself. Of course, computer now, everything is digital. You convert everything you type into a number, and everything you see on the screen comes from a number that will define the color of on the screen, every pixel. Inside, there is new math called binary logic. Is done on ones and zeros only, and you can do everything with it. So that's what got associated with digital information, the computer language. But anything that's discrete is a digital information. Symbols, notes, letters, all of this. So the concepts we're going to think about, that natural selection and mutation. Natural selection is basically the mechanism where by reproduction the offspring will be favoring certain direction more another and will continue. And mutations, which is problems that happen in this reproduction that makes the next species either have a inability to continue on a certain direction or die, or have certain difference in the shapes. All of this is not going to be my focus today. It's very well established. There is natural selection, there is mutation. I'm not arguing here. The argument here is that does this account for the origin of life? Does this mechanism can explain how life started? That is where the gap is. Origin of life happening by chance is very improbable to the highest degree. We'll give you an example of it. But just everything came along by chance. Time from the beginning of the universe or the beginning of the presence of creatures till today is not enough to bring every single detail in every single creature. For natural selection plus mutation to occur, reproduction must be in place already because it's natural selection of the offspring. So it's a mechanism that starts operating after reproduction. There's already a reproduction mechanism. So how can it explain the origin of life? We're going to tackle this. If life hasn't started, how can a mechanism that takes effect after reproduction is in place, how can it explain the origin itself, where reproduction hasn't happened yet because there's no life yet? There's no offsprings yet. So here is a basic challenge, a mechanism that takes place when reproduction starts, biological reproduction, offsprings. How can it account for the very beginning of the existence of life? It hasn't taken effect yet to begin with. So that's a very basic tenet of it. Necessity, self-organization, biochemistry is a very, very important concept we'll do today. Chance and necessity came from a French author. That's why it's used chance and necessity. But in fact, necessity means there's certain laws that maybe guided the chance to make the really life get guided much faster. And we're going to challenge this also scientifically. Elementary models of life always at some point make a leap which requires specified information. So there is some challenges to the life. Reproduction happened before the presence of the DNA, because the DNA is the best explanation for the reproduction. There's certain information. When it divides, there's cells division, and then you can have the traits carrying on. But when we say, okay, then DNA has to exist to begin with. Where did it come from? Where did the information in it come from? It says, oh, before it, there is a model called RNA model. At any claim, if you're not savvy enough, and sometimes with a discipline that deals a lot with information, you will get duped that there is a certain step that's claimed and it requires information at that step. That's in the RNA models, but I will not cover it at all. It's a big topic, but I'm reading about it. But my background is not biology. It takes longer to to understand this. I envy you sometimes if you have that background. So we need to be trained to spot where the leap or the step that was said in the middle of the writings, where actually information was needed. And that's where you get fooled, exactly where you get fooled. I'll show you examples with some systems as we go along. So as I said, RNA model for the origin of life is an example of this. Information enigma, or specified complexity, or protein 3D shape, order of amino acid and protein synthesis from mRNA. We're going to cover this, and it all comes down, where does the information for all of this to happen, where does the information come from? Because it's not a short piece of information. And these detailed systems are extremely complex. But we'll get deeper into them. So it's two main things. You get fooled when certain things are claimed, and at some point information is needed, but they don't mention it. The other thing is that there are certain very complex systems in biology. For them to operate, there is a lot of information that have to exist all at once for it to operate. And future research is to focus more on this aspect of information and the RNA model, because it's the challenge. So let's start with the top. Darwin's model, survival of the fittest via natural selection plus mutation. It is ingenious that he is using the present to understand the past. It's very, very intelligent. We shall use the same approach, in fact. We shall use the presence of systems that we find in biology, and we'll try to conclude about the past. So it's basically what forces now are in motion and prevailing to give us understanding of the past. What things are now working? How do these systems work? And we'll try to see it and try to conclude about the past. Dawkins said, this is now evolution, we use evolutionary theory to remove God. Darwin gave us design without a designer. This means that natural selection can mimic the power of intelligent design without an intelligent design. There is no God. Needless to say, natural selection is a blind process. It does not have foresight. It is not goal-driven. It is just a process at work. Please note this down. Natural selection does not have foresight. It's not like a control system like a GPS system where I know where I'm going and I'm solving backwards from where I am to get there. These are intelligent systems because goal-driven. Natural selection is not goal-driven. Natural selection is a blind process, has no foresight. So this is the very first concept. It's a process that doesn't know what we're designing. It just goes. And he's saying it can mimic the power of intelligent design without intelligent designer. It is a completely unguided process. In other words, as Meyer phrases it, Stephen Meyer, I rely on him a lot in that research, that Darwinian intelligent design gives us life that looks designed, but it is through a purely undirected natural process, which can induce that appearance without it itself being designed or guided anyway. I just want you to read this paragraph again, because it's the claim and the answer to it. Natural selection mimics the power of intelligent design without the need for an intelligent designer. It is completely unguided process. In other words, Darwinian intelligent design gives us life that looks designed, but it is through a purely undirected natural process which can induce that appearance that things are designed without it itself being designed or guided anyway. So I have to challenge this in an engineering approach, looking at biological systems and see the amount of jumps when they try to explain them in biology. And I will explain to you some terms called reductionism, the gaps in the explanation, the hypothesis that they claim, and there are so many details in it that you don't get explained how. They just throw it on evolution. For a design system, these are crimes. It's just an engineering crime because when you make a statement like this. So, but I'll take you with examples. Okay, to move a little bit faster, in the 19th century, people were enamored by adaptation, how fish have gills, sheep in Scotland have wool, how breeders can direct the offsprings of sheep to have certain traits by genetically marrying, breeding sheep together, and so on. So Darwin claims that nature can accomplish what the breeders are doing. So nature actually can direct in a certain direction. So breeders take two types of sheep, breed them together in order to get a certain trait in the offspring. And if you continue this like for 2 or 3 or whatever generations they decide on, that trait stays very, very strongly because they find it's maybe a lot of wool, economically is very viable, whatever the reason is. But the breeding is directed by the breeders. So Darwin claims that nature actually does that for us. It directs where it's going, which is completely questionable because as we know that natural selection does not have a foresight, does not have a goal to. The breeders do. They know what type of trait the sheep that they want to have, lots of wool, lots of fat, whatever it may be, they wanted to have it. So they breed in that direction. Nature doesn't breed in that direction. Nature doesn't have a goal. It's a foresight. It's not a process with goal in the mind. It's a crucial question, both of them. Is adaptation the only appearance of design? That fish have gills and scales? Is that the only appearance of design? Or there is so many things that have design in them? And if not, does natural selection explain all other forms of design? So these two questions also keep them in mind. Is that when people talk about design, they talk about it very lightly. And I attribute this really because of the discipline of biology and biochemistry, and these sciences do not have the rigor that the discipline of design on the side of engineering and the analysis to get something working out, the details of it, is just absent. So when the word design is said, it's just thrown out without any rigor in what you mean, what you're talking about. That's not at all in the engineering discipline. The design has specs, has measurements, have the order of things, have things fitting together, have things operating, have sometimes feedback system in order to adjust the process automatically, like the autofocus of a camera, for example. All of these are amazing design, and you see them in biology and it just says, oh, I'll show you an amazing example how it gets explained, and I hope it would hit home. Recently, as stated by Meyer, there is an increasing call for a neo-Darwinian theory that can match the new scientific of more and more evidence for the presence of design in biological systems. This is the challenge that will always plague, and it will become more and more harder to answer. Thank God for the science, because of the discovery of the more details in every single system, specifically the cell, and what the cell does, that made it now the gaps you have to cover to swallow the design gaps that you want to jump over are bigger and bigger and bigger. But because in order to publish in scientific community, you have to value an evolutionists, as long as you put the word evolution there, find some proteins that are common between this and the ancestor, it's called reductionism, by the way, you get home free. And this is a crime. This is really a crime, because you're not doing science. There's so much explanation you have to do in terms of design, and you're not. And you're teaching this in books. I actually have passages from the Campbell book today. I scanned it to show it to you. So in 19th century versus today, 19th century, the cell is believed to be a globule of plasm, and hence science was satisfied that Darwin's theory was the full explanation. It was then the view of the cell is completely different than what the cell is today. And also the chemical evolution was fully supported by the Darwinian theory. We'll talk about the biochemical destination today, because we're going to challenge it very, very elegantly. That in the tree of Darwin, of life of Darwin, which starts from a single cell and gets more speciated till it becomes all the phylum at the top. In fact, in Darwin, you have to watch Darwin's Dilemma. In fact, the Chinese scientists, not religious, they are saying this tree has to be inverted. The number of phyla appearing all at once is very, very, very many a number at the same time, that it's undeniably the tree of Darwin has to be inverted. And these are clips from Chinese scientists that are claiming this. So anyway, at the very base, there is the origin of the first form of life, then it branches till we get at the top all forms of. This tree is drawn by a person. This is his hypothesis. It is not a fact, it's a hypothesis. And Darwin wrote in The Origin of the Species, he said, if the Cambrian explosion, which is the presence of so many species in a very short time, is proven to be correct, and then it proposes a very serious challenge to my theory, because his theory is a very gradual process that leads to the different species at the ends of the tree, or the branches of the tree. So Darwin hypothesized something. A single cell from it came everything by the process of natural selection and mutation. And now with discovery, we're discovering things appearing with so many details in a very short period of time that challenges his description. So this plus design plus other things, there's going to be, I think, 6 or 7 things I'll give you at the summary at the end as challenges to the theory of evolution. Darwin, however, did not attempt to solve the origin of life systematically. He just said, here is a cell, that's the origin of life. Systematically means in detail proving that cell came on its own. That's systematic proof. Darwin described the natural selection process and hypothesized that it explains life without systematically explaining how exactly life started. Again, where biologists need to learn from design people how to explain something that shows design. However, Darwin gave us the genius of how to look into the past using the present. That's definitely, it changed science. Fred Hoyle, I think his contemporary, a geologist contemporary also, that claimed this, and it stuck with both of them very well. Lyell, not Hoyle. Fred Hoyle is the chemist. Till this, and this is just the structure, not what it carries inside. The double helix, Watson and Crick in Nature, I think 1952, you're more expert than this, far than me. But they discovered that there is a certain cell that carries a certain odd shape, and but they don't know what it carries. What does it mean? What is that order meaning? But it carries information. 1953 in Nature. From the past to the present. So this is just the discovery of the shape, the double helix, and now coming today. The molecular biology revolution happened via the discovery of the structure of the DNA module, this double helix, by Watson and Crick. It was discovered that the DNA molecule does not only have the double helix shape, but more importantly, it carries, it carries sequential arrangement, an order of binary information. As I told you, binary, think about as a book. A book is a binary information. Information that looks like letters in a sentence where there is an ordered sequence information being conveyed. Proteins carry, among many others, the following jobs in the living organism, because we're going to talk about the protein synthesis, so we're going to focus on one of the functions of this double helix carrying sequence of information. It acts as enzymes, catalyst to reactions, acts as parts of machines that carry out different functions in the body, actually carry out the functions of producing enzymes or creating the amino acid sequences, as we'll see in the protein synthesis. Proteins are made of amino acids. The sequence of these amino acids is unique to every protein. Proteins are made of amino acids. The sequence of the amino acids is unique to every protein. But also a protein molecule, this is the part that got me, you learned about it very recently. The protein molecule has a three-dimensional structure, which defines also the function which that protein will perform. So it's not only the sequence of the amino acids. So a protein, actually I got here something to, if amino acid we're going to show how it's connected together, but I have here like a toy. So in order to produce a certain sequence, you have this order. So this order, let's say each one is an amino. This is a protein. This is hardly anything useful, as you all know. Proteins are usually longer sequence than this, but this order is very important, and it's related to the function of the protein. Not only the order, but also the shape. This is a straight line, but this protein has to be shaped in a 3D structure that will be relevant to the function the protein will do. Now, when you come and tell me this 3D structure, evolution decided with trial and error how to find the correct 3D structure, it's in fact, it's a crime even as a scientist to say this. Don't throw it on evolution. Don't throw it how a biological system has the ability, not only that the order has to be correct, but the part that is absent from us, how it gets folded in a shape, a unique shape. A unique shape in 3D has infinite number of combinations, independent of the rotations, but the location of each point in 3D on the protein, in order for it to have that shape, is exactly when you design anything in a car. It cannot just be a piece of metal. It has to have the tolerance, it has to have the shape for the function it will do. This cannot be thrown on a process that doesn't have foresight, or a bunch of proteins getting together, and with time they will figure out the mechanism that will fold another protein in a 3D shape. So here is when you're getting the sense of there are certain things that are written in your books, in the biology books, and you read it and you pass over it. But the discipline of engineering, when you read this, you can't pass over it. Where is this? Who folds this? And how does it get the information to fold? It's like when you go to a machine shop on a lathe and they build for you something. There is a person that's standing, adjusting all the precision in order to get everything correctly, and it produces a machine part, which will be used in something else that has maybe thousands or millions of machine parts, an aeroplane or a car. Anyway, I can't belabor the point anymore, but please, if somebody finds a scientific explanation of how proteins fold other proteins into three-dimensional unique shape, it would be good, but we want something scientific, not maybe or perhaps. The order of the amino acid as well as the 3D structure of the molecule are two main, the order and the structure, are two main attributes that defines the protein and the function it will perform. It was discovered that this function fit mechanically with the protein as hand and glove. So let's get into the protein synthesis. In order to produce a protein, we'll find that the DNA has information how the protein will be formed, and the protein is life basically, because everything, you know more about this than me, I can't say anything further, but as far as I know, everything is protein, but they do different functions and they interact with one another in order for to perform this function. So information is not sufficient to produce a living organism. Processing of this information is needed. So not just the information, but the way to use it in order to produce. How to execute the information on, which is how to create life, creating a protein, because we're about the origin of life. So how a protein is created is important. So you need information, and you take this information, process it, do something with it to produce at the output a protein. As we agreed, we're trying to understand the origin of life, and the origin of life is proteins. So what we need is to build this protein. So protein is the functional element for any living organism to function. The conversion of DNA into protein relies upon a software-like system of commands and biochemical codes. This command-based information processing system is in many respects identical to the types of commands seen in computer software. Bill Gates said it's like a computer program, but far, far more advanced than any software ever created. Richard Dawkins agrees the machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be or could be interchanged with those of a computer engineering journal. There is software being executed in your cell, and that software, some machine executes it, and the output of that code is the sequence of the amino acid that is the protein, the unique protein that that piece of code, when executed, produces. Then another machine takes it and folds it mechanically and then gives it to the function it's going to do. This is a factory, a well-designed machinery factory. So this is the process of how it happens. I think if we watch this movie, I just need to end sharing and then get the sound from the computer in order for you to hear it well. So we're going to share. We're going to share, and then, so I'm going to play it. If you can't hear it, please let me know.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system.
Can you hear it?
Yes, we can hear it.
Okay, thank God.
The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled. by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns.
Just to explain, or not explain, it's basically, there's so many things working here that have to work correctly, which is, where does it start and where does it's a piece of code. That piece of code is going to be taken out of the nucleus to be executed, to be run. Executed means not killed. I mean, it's executed. When a computer executes code, means it does the code. So here is several things. How to know where to splice it, where it starts and where it ends. There's a beginning of code and there is end of code. That's a computer programming starting the code, or a sub-function. If you do any code, anybody would be familiar with these terms, that here it starts, here it ends, or here it's the beginning of it, and here is where it returns to the main code, or if it's the code itself, where you reach the end of execution. And besides all of the mechanism of cutting it and sending it and the separation of the DNA, all of this, people with the background, I don't have the background on it at all. Michael is there. Ask him till you very, very satisfied about anything else. But when I look at this, I look at it very, very detailed. If this is off by one, it won't work, because this red part, the mRNA, also has parts that will be taken out, will be spliced out. So you need a machine of proteins will come and take these introns out, and the red, which has the continuous code, so you splice it and then you put it together, and still the code is continuous. I mean, I don't know what computer code, you go into the functions and take part out of it and it performs correctly, and where to know how to splice and to stitch it. So please, when somebody says nature found a way, the amount of intricate parts that have to work together all at once for this to happen is as a person sitting down to write code that the computer will do something intelligent. So I stopped here at the slicing. The beginning, it has a beginning of code, an end of code, and what's in the middle is actually called codons. And the codons will be matched 3 at a time. Why 3? Well, who decided on 3? Why not 2? Why not 4? Why not 6? So I don't know, when you circumvent that design by, oh, science will discover in the future the mechanism nature found the number 3 is the best. Even if you say it's the best, how did you measure it against the worst? So how do you know the other was worse? There has to be a feedback into the system to know this worked, this didn't. So, and we know that evolution has no foresight, has no goal-oriented. It's just a blind process, as even at the very beginning said, it's an unguided process. And it's just a system, a tiny system, not tiny, I mean, our livelihood is based on this function reliably. Without, if evolution is true, and this is a trial and error, how are you going to try and error proteins? How will the species live? How will the organism live if the protein production is trial and error? So we'll continue. So we're gonna do from the splicing, and then this will go and become executed three instructions. I'm using computer jargon because it is a computer. 3 instructions at a time to give you the output of the code, which is the code is encoded here in this mRNA.
In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA.
Okay, in computer jargon now, the first instruction of the code, and there is a beginning of code, this 5' and 3' that got put at the end. So now we're going to execute the code 3 instruction at a time, and each instruction has a code that is formed by 3 letters, and it gets matched by a unique one, which is the tRNA, and the tRNA carries, so the mRNA is executed 1 instruction at a time. Each instruction is a 3-letter code. That's the code for it. That's why it's actually the word codon. And the matching one comes. This is chemical affinity that between them. That's why it decides who joins to what. That's completely chemistry. But these 3, the A, U, G, is what made the UAC connect to it, carrying the specific amino acid. And this gets repeated, and you get the order. So the first one comes, and then the first one, and then the second execution, then another one connects, and the third and the fourth, and so on. That's basically the code in the mRNA after it was spliced by a protein and put together is going to give you a protein that the body needs or a process needs. This is your expertise. But this is where I stop, and I have to, in awe, to say, how can this be explained? So please, this is where, as I'm telling you, the claims of today, accounting for the origin of life using evolution, is being challenged by these type of systems. And I'm going to get into more other type of things shortly, but this is one of the very highlights of question marks. So we're going to execute that code 1 instruction at a time, and every instruction is a 3-lettered word that's basically called, we call it in computer language, the machine code, or the instruction that needs to be executed, and the matching tRNA is the execution of it, and it connects the amino acid together to give you the whole chain.
After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. a release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
I hope this does it some fairness in seeing how complex it is. I just wanted to see how many machinery at work. Of course, you're familiar with it with biology and biochemistry major. I'm ignorant of this completely. I left biology since 11th grade. But what jumps out is what does not jump out probably with the biology and biochemistry background, because what jumps out is a machine, is a computer. Flat out computer. It has code, it gets executed, has a beginning and end of code. The code has a certain result, it's accurate, it's repeatable, and this accuracy is not up to chance, because if it were, the organism will not live, because you cannot do trial and error. Oh, this protein is wrong, or how, again, even if it's true, what mechanism knows it's wrong and feeds back into the system to modify it to get it right the next time? So there's so many claims that, as I said, there's certain engineering discipline. One of them is control systems, which has this feedback loop in order to feed the information back from the output to the input to modify the system. Machine learning, there is so many aspects here of disciplines that started very rigorously and is appearing in biology. Since the discovery of the DNA, biology became engineering because it's a digital system. And I'm going to show you more. A very big discipline of engineering in the DNA. We'll get to it very shortly. But before I leave this, let's see somebody who computed the probability of, here is the probability of getting the correct protein in wrong ones by chance. So basically the system is, the DNA opens, it gets a piece of code, somebody above it tells it we need this protein, so it goes, opens, gets you this piece of itself out, which is the mRNA, and there is a beginning and end for it, and then it gets spliced, again, with another protein machinery, gets spliced and stuck together where the code is still seamless, no interruption, no error in it. A beginning and end gets put on it, and then it goes to execution, one instruction at a time, and each instruction has a code of 3-letter word, which brings the execution of the instruction correctly in another machine that knows how to put them together, and the sequence gets done till you reach end of program, and it finishes. It's not infinite, and the length of it is matching the length of the polypeptide or the protein, and the sequence is needed, and then it says at the end it goes to get adjusted again. That adjusting is mechanical engineering, folding it in a way for the function it will do, not just the sequence but the 3D, so the order of the amino acid and the 3D shape of the amino acid. So comparison to source code, this is really, I talked about it enough. I'll just show it by pictures here. The inside of a processor is exactly this. Fetch code, decode, fetch an instruction, decode what it is, executed. This is called the fetch, decode, execute cycle of any microprocessor to do code. That's basically what Bill Gates and Richard Dawkins said. This is a computer at work. Just if I need to go deeper, this is how the arithmetic logic unit and how an execution of code happens in a computer, but will not need it. So where evolution has challenges, let me not say fails, but the challenges where evolution has challenges when it claims that it is independently the origin of life. The DNA has information. This information is ordered in a certain sequence. The amount of items that are ordered is so large, 3.2 billion. This is what Francis Collins, who is the father of the Genome Project in the 90s, was able to tell us the order of the DNA. This is different than Watson and Crick. Collins, under Clinton, actually got a highest scientific honor from Clinton, and he wrote actually a book called The Language of God. Francis Collins is the father of the Genome Project. The Genome Project is the project of labeling the order of the human DNA. Now we know what our whole book that produces a human looks like. So the information it's not small. Actually, he said it's estimated if this is written in a book and needs to be read with a person reading 3 words per second, it will take the person non-stop day and night 32 years to finish the book of the DNA, of the human DNA, non-stopping reading 3 words a second. The amount of items that are ordered is so large, it will completely be improbable to have time using self-organization evolution model, trial and error, to rearrange them till you get a successful functional intelligent system. I would say to get successful one protein. This system has the process, to say the least, of irreducible complexity. Irreducible complexity. There is a reducible complexity, like the complexity of a sodium chloride crystal, for example. It's complex, but it's reducible to the first element of it because it repeats. That's reducible complexity. This is irreducible complexity, means you cannot spare one part of the machine and it would still work. Everything is needed at once for the protein to be produced. So that's called irreducible. You cannot reduce it to simpler parts and say this gradually evolved into the more complex part. They all have to be there for the protein to come out. Everything, which you know more about it than me because you know their names, I mean, with your background. But for you enough to know that the amount of proteins needed for this protein production to happen is large enough, and they all have to exist. So inability to explain complex processes. Combinatorics, we'll get into it now, the combinations challenge to evolution. Gaps in the species, I will not cover it. It's about the evolution trees and the gaps in it, but I would direct you to the Darwin's Dilemma YouTube video. It's available on YouTube. So I want to give full credit to Michael Hanna, who's presenting today with you. He's the one who brought to me these paragraphs from the Campbell biology book. And this is, I'm quoting the book. The molecular, after they explain the process we just explained, that the translation transcription of the DNA to produce protein, or the protein synthesis process. The molecular processes underlying the protein synthesis seem inexplicably complex. Although we can describe many of them, they do not make conceptual sense in the way that DNA transcription, DNA repair, and DNA replication do. Protein synthesis also relies heavily on a large number of proteins that are bound to the RNAs in a ribosome. For people who are ignorant like me about biology, just take this at the surface level. There are so many moving parts that splicing, where the amino acids are connected together. All of these that people with the background of biology are very, very familiar and expert at. What to take home is that all of the machinery has to exist. The protein synthesis relies heavily on a large number of proteins. The complexity of a process with so many interacting components has made many biologists despair of ever understanding the pathway by which protein synthesis evolved. Because you see here is the dictatorship. I want you to see how the biology books are written. Because it comes from the top, it has to be evolution. And now the energy of science is going into finding the evolutionary process to explain this. We're wasting time. But I cannot say clear this because it's a faith statement, because I will say God made it. I'm not going to claim God made it. But your scientific system starts with a premise. The premise is one hypothesis became a fact. It's evolution only. It's all of our time now has to be dedicated to find the evolutionary path where this machine came from. And that's where all of the research, all of the energy, all the grants, all of the taxpayer money is going, because it started with a faith. That's a faith. It's called the faith of evolution. Because the scientific stuff we're presenting here is a challenge. I'm not saying therefore God exists. I will not say throughout the presentation. I'm saying there is a challenge. So again, thank you, Michael, because he directed me to their statements, and we'll have more statements from the book. My daughter came into junior high, junior high school. So I have that Campbell book at home now. I'm so happy to have it because I can get now more of these statements that's really, I cannot be sarcastic in all due respect. It has major holes in design. So the despair of even understanding the pathway by which process senses evolved. It has to have evolved. That's the faith. It has to have evolved. This is, by the way, a judge in Arkansas ruled that everything scientific has to be by methodological naturalism. That's an elegant way of evolutionary theory. It is claimed to be the definition of what science is. And I can't remember the case, but I have it. It's the Board of Education versus someone, and the judge had a ruling. Legally, you cannot do science unless it's by evolution. Unless evolution is the background. That is not science. This is exactly a police state. It says that science is limited to providing strictly natural, especially physical, explanations for strictly natural, especially physical phenomena. Sure, but the claims that are doing now are metaphysical. Methodological naturalism usually appeals to natural law. Those who practice science are told that they must limit themselves to the rules of what counts as science, naturalistic causes, and natural laws. So the other one, because we're trying to insert God into the schools, and it's like a way to do it. Please present intelligent design as another hypothesis that's not plausible or not good, but present both. But no, it's only one that's presented. Continuation here. As discussed in chapter 1, early biological reactions probably use RNA. Here is now where we're going to come to all terms of what we call assertions or uncertain assertions. Probably, may have, maybe. That is not science. That is a hypothesis. As discussed in chapter 1, early biological reactions probably use RNA molecules rather than protein molecules as catalysts. In the earliest cells, tRNA molecules on their own may have formed catalytic surfaces. These are all designed again. How did it form catalytic surface? How did the surface look like? What type of proteins it takes to form a catalytic surface? That allows them to bind and activate. Again, which amino acids were activated? How? Specific amino acids without requiring aminosyl tRNA synthase enzymes, which is needed now for all of these processes to happen. You see here, trying to create a pathway to explain this without somebody put the information and somebody put all the machinery together. Because we need both, as if you remember, the information and the computer that will process the information. The information and the machine that will execute the information. The information we found in the DNA, and the big question now that I want you to ask always ourselves, where did this information come from? That's a 3.2 billion letters long. And the machine that executes it, how are all the components found at the same time, simultaneously, in order for this information gets converted to what this information is intended to do, which is in our case here, just the protein synthesis. Another question, what is the machine above this that tells I need this protein now, where it makes the cell go and produce this specific? This is another, what we call it, a higher level of control that decides based on other input, we need this protein now. So it's another level of intelligence, of decision making, that this is working as a slave to. In control, this is called master-slave process. There's some master decides on this, and then the slave machinery says, okay, I know how to do this, let me give it to you. But there's a decision making on we need this protein. more with the biology. I'm quoting the biology book, and Michael has provided me with these quotes. Excuse me. Likewise, RNA molecules may have served themselves as the entire ribosome, folding up in complex ways. How? What shape was who decided on the shape? Complex ways to generate an intricate set of surfaces. Again, a very detailed design machinery that both guided tRNA pairings with mRNA codons and catalyzed the polymerization of the tRNA-linked amino acids. So please, in this sentence, when you read it on its surface, it's completely hypothetical. And when you dig deeper in every single statement, what is required for what they are telling here is another layer of complexity that is full of hows. But this is where the research is headed with all the money, because we have to fit this to be formed by itself or with an unguided process with no designer. On its own, it found a way. So please, this is what I notice. In mathematics, there is a field of probability. It gives a certain number. By the way, probability is the underlying science that makes your phone works in transmitting data from one place to another, voice data. How this is done is two fields called communication theory and information theory. Both are built on digital information. Were invented, as we'll get shortly, by Claude Shannon in 1948. When you find the word possible, it is not equal to the word probable. You get fooled by the word possible because everything is possible. Well, what is the probability number that this event would happen? If it's 10 to the power minus whatever, something bigger than 10, then the probability of this, the likelihood of this event to happen is almost zero. And we can call it impossible. But because you find in your books the word possible, everything is possible. And you take it home, oh yeah, it's just a matter of time. Wait a minute, the probability of this to happen is very, very low, because as they say, the complexity of the system they claim that will be discovered have systems under that, and they make decisions. Functions as means I built something to function. I'm trying to highlight to you the sentences that are used are very, very foolish and unscientific in another field, completely unscientific. So there is a big difference between the word possible and the word probable. The word probable is the one to be used. How probable is this to happen? Almost zero. But when you put the word possible, okay, you go home happily, it's possible. That's not how science is. So be careful between the two words. Not everything when they say possible is possible. If it's very improbable, the likelihood is very low, then it's almost impossible. Continuing on the book. Over the course of evolution, individual proteins have been added to this machinery. Well, at least there is admission here that it is a machinery. Who adds it? Not who, like a person. What another decision making that decides to add these proteins to function as what? To do what? What is the lacking in this machinery that decided that we need this, and another machine or another process said, okay, here is the protein that will make you do this. This is, I keep repeating this to get the concept that what is said in biology books is laughable. Each one making the process a little more accurate and efficient. Okay, who measures that it became more accurate? Why it's not less accurate? Somebody must have measured outside the whole process and found, oh, this is good, let's keep moving. But evolution is unguided. It has no foresight. So how is it decided it became more accurate? Who decided it is more accurate than before and efficient? Or adding regulatory controls. That's a whole system called called control systems. That is really, really in this feedback and stability and things that we got all subjected to in undergrad and grad school and further. In this view, the large amount of RNA in present-day ribosomes is a remnant of a very early stage in evolution before proteins dominated biological catalysts. I'm writing here, what the heck are you talking about? Because I'm not being sarcastic, but I'm saying what you say here for students, please present us with papers that the measure of the machinery that found we need to add proteins and the measure of accuracy and how biology on its own unguided measured the accuracy, decided to to add it in that direction. These proteins got produced in this way to make it more accurate. Please give me the rigor, or at least the process of science that is going to prove the existence or the working of this machine. None, really none. But I might be ignorant. You guys, if you're in that field, please, I would love to learn more and more about it. So we'll get into another challenge. So this is a very complex system, and there's another challenge, which is the combinatorics. Let's say that we'll produce a protein that's 150 amino acids long. Dr. Douglas Axey, who's a molecular biologist, PhD from Caltech, and he works at Cambridge University, estimated that for every DNA sequence that generates a short functional protein of just 150 amino acids in length, there is 10 to the power 77 amino acids arrangements that will not fold, will not have the 3D shape into a stable three-dimensional protein structure capable of performing that biological function. So we get one right in 10 to the power 77 wrong. So what is the probability of getting it right every time? There must be a repeatable code that generates it correctly. That's why the chance or things on their own happened became now as a completely implausible concept. One correct sequence for every 10 to the 77 power incorrect sequences. To put this in perspective, keep in mind there are only 10 to the power 65. This is 10 to the power, it gets big quickly. 10 to the power 65 atoms in the entire Milky Way galaxy. This again from scientific quotations. You as a biological creature are constructed from cells, which in turn are constructed from proteins, for which DNA gives the coded building, as we said, instructions, and another protein folds it in 3D shape. Searching that space of combinatorial possibilities for just a single protein, which is 10 to the power 77, that space of 10 to the power 70 is far beyond the resources of even life. Whole history will not be enough to get that one protein correctly. If it's this is number of things you have to search in 10 to the power 77. In the past 3.5 billion years, only 10 to the power 40 organisms have ever lived. Each such individual organism represents a potential trial of the creativity of unguided evolution. Even if we assume, which is very generous on our behalf, that random mutations gifted every single organism in Earth's history with an entirely new peptide sequence 150 amino acids long, then that figure of 10 to the power 40, which is 10 to the power 40 organisms, is still just a tiny fraction of the 110 trillionth of the earlier figure, 10 to the power 77. So 10 to the power 40 is all of the biological organisms. Let's say we got them all correctly, still forming one protein in 10 to the power 77, which is the 10 to the power 40 has to search in the 10 to the power 77. That's a humongous space. It's a fraction of that space, 1 trillionth of the fraction. So which means the search space for things to happen in chance is impossible to figure out in the lifespan of the world for one protein, which quantifies the space to be searched to produce a lone functional protein. Sufficient time is just not available for Darwinian evolution to do the job, not even close. So we have now the design challenge, we have the combinatorics challenge, which translates to time. Chance is not enough to do it. Third one is jumps or gaps in the species. I will direct you to that video, Darwin's Dilemma, and I will skip that part. But there is major jumps in the tree of life, and this is because of what is called the Cambrian explosion, which is a lot of species appeared in a very short period of time, which is using this protein mathematical analysis poses a very big challenge. That time is not enough for you to have your chances coming correctly. Example of the 3D structure we talked about, the sugar has a certain shape, or the enzyme that breaks the sugar, So the protein that breaks the sugar is actually folded in a 3D shape that it creates a mechanical tension on the bond of the glucose and fructose bonded together in order to break the glucose from the fructose. The enzyme that breaks it is mechanically breaking it. It's actually physically pulling them apart. So the protein, this enzyme is not only formed with the correct amino acid, it's folded in a way that it's a machine that when the when the substrate enzyme substrate complex sits in it, it actually creates a tension on the bond that it breaks it. That's an example of a mechanical design that is needed for this protein to do this function. And this is consistent because obviously you're breaking sugar in your body. So where would be the trial of error available and the success of the species to continue if this is not working correctly from day one? How would you generate functional organism if this is not working from day one? And what is the biological system that if it's not working goes back and fixes it? It's so an example of such phenomenon is the breaking apart of the two-part sugar molecule. We can see the magnificent structural fit between the protein. That's what's called the hand and glove structure, molecule and the sugar molecule. Information. DNA molecule has an instruction that encodes how to build. It's a book. When you execute this book, a species comes out. Ours is 3.2 billion letter long. A human comes out. They have the exact function of a computer code that when executed produces certain intended outcome. We covered this. This is just a recap of the pictures of it, the transcription, translation of the protein synthesis. Here is the breaking of the DNA molecule and the mRNA, and then the execution of it with the tRNA, and you form the peptide sequence here, which is the protein. And then later on it gets folded mechanically. Same, another depiction of it with the mRNA coming from the DNA being executed, and then you generate the protein, and the protein again gets mechanically folded. Okay, so I think this is just a re-hitting the point because of the number of moving parts that have to work correctly. All are the same. So the additional information here of these chemicals, the G and the U and the N and T, it's copied, it's copyable. It gets transported. It executes as a computer code to direct the construction of each protein, the order of the amino acid, as well as the 3D shape of the protein. It's very specific. It's very large. The construction is done in a chemical factory called the ribosome. The amino acid folds into 3D structure in another factory. This is just to hit you in the mind the number of engineering design that needs to be done correctly for the protein to do its function. From the conception, and besides the layer above it to produce that protein, which is, here is the code, as if it's a computer coding what instruction does what, but I will just show you images because I belabored this point. And here is the code of Richard Dawkins. Again, the machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer engineering journal. So as we said, as the computer is a fetch, decode, and execute. So this is the fetch cycle, which is we know what the codon is. We decode it, what tRNA needs to come and connect to it, which is this part, which part matches it, and execute, which is, okay, we're going to get out the amino acid. DNA carries information in the form of an arranged sequence to form the protein in a specific amino acid order, as well as structure. The ordered sequence stored in the DNA acts as a computer code that generates a certain order output, as well as a CAD/CAM, computer-aided design, computer-aided manufacturing tool that generates a 3D structure. So also the machine that folds it mechanically is another thing that I don't know if there is anything written about it in literature. How does it fold it mechanically? You might know how does it bend it, how does it form it that it takes a certain shape. So the DNA enigma is not the structure of the DNA, not that it's helix. That's not the magnificent part. It is magnificent. Not where the information resides, which is the form of these connections or affinity between these the G and the C and the U and the A or the T. This is not the magnificence is not the what we call as the hydrogen bond between them. Yeah, the helix is magnificent. The bond and the certain to have affinity, another to have another affinity. That is still not the miracle of it. So the enigma is not what it looks like, not where the information is, which is in these bonds,
أبونا
what
جريجوري
information does.
أبونا جريجوري
Yes.
قدسك my mind ما يقدرش to occupy all these informations. It needs a different DNA and RNA.
Should I stop? Well,
I believe it needs we ask good questions.
okay,
So
this is the last.
we can digest what Godsak said already.
Okay, بس there is an important.
Because this it will give us a different substance. If we continue like this, DNA والRNA بتاعنا مش هتقدر to.
طب I'll hit just one point and then
Sure.
we'll get for questions, and
Sure.
في another part maybe we'll have it for another conference, but let's just hit this part. So the enigma or the miracle of the DNA is where the information came from, and I will give you a very simple example. So let's look. I don't know if you see behind me here. Do you see these letters behind me on this box? Can everybody see it? I just need an answer.
Make yourself the instead of the PowerPoint presentation.
So we end.
That's a little bit better.
Can you see it?
Yes.
Maybe I can bring it closer. So there is each one of these letters has what in it?
Magnet.
A magnet. So consider this magnet is the chemical that holds the DNA strands together. Like if I share with you, I'll come back to, but I just want to show you one picture. Okay, so in this picture here, the chemicals which form the helix are held together by a nitrogen bond. By a hydrogen bond, and they are held together to the helix walls by a nitrogen-based chemical. So the bonding between these two is like the magnet. So the miracle is not that they are held together or not the shape, but the miracle is exactly as I will show you here. If I stop the share, consider each two holding together is like the magnet holding to this piece. The property of magnetism, الخاصية المغناطيسية, the property of magnetism is what holds the letter to this. That's like the hydrogen bond connecting the two chemicals together of the DNA. Now, what is the miracle? It's not in the magnetic field, it's not on the hydrogen bonding, it's not in the shape of the helix. The miracle, or the difficult to explain here, is if I do this. I'm going to form the letters in a certain form. Can you read what's written? Is it clear? So what happened here? Someone took this ability of the magnetic field, or in the DNA case, the hydrogen bonding, and he wrote with it a long sentence. And that long sentence functions in a certain way, which is what I did here. I took the letters and I formed it in a certain order, ترتيب معين, a certain order to form I love you. This gives a meaning. That is what the miracle, or this is the enigma. Enigma يعني الموضوع الفذ في الدي ان ايه. مش شكله, not the shape of it, not the bonding that's happening, but the question is very, very big. Where did the information in it come from? Because this is what someone did. He took these chemicals and he formed with it a sentence. He took these chemicals and formed a sentence. This sentence is not because of the magnetic field. I love you does not look like this because of الخاصية المغناطيسية. I love you is formed like this because I use the magnetic property to write the words. So I gave you a very simple example. There is more stuff, but a very simple example here to show. Here is a mechanism that's very, very elegant. It has information in it. And the question of it now, where did this information come from? Because this information is what makes proteins, what makes us live, and the protein making process is extremely, as we said, complex. So the origin of life question to evolutionists is the origin of information. The sentence is not sticking to the background because of the magnetic property. The sentence look like this. Sorry, the sentence does not read I love you because of the magnetic property. The sentence reads I love you because an intelligent agent used the magnetic property to write the words I love you. And time will not permit, but were more examples of how in the biology books there is examples of very complex systems, and they do something called reductionism. What is reductionism? They say, oh, this has this number of proteins. Here they are 19 proteins. And another ancestor of it has, sorry, this has 21 proteins. We found 19 of them in their ancestor. Therefore, it must have come from it. So they do reductionism as if your microwave and your car are made from the same metal, then they must have come from the same origin, from another piece of metal. A lot of this is used in your biology book. Please, it's called reductionism. When you use the same components, the same proteins, they found it in one species and another, they relate them only because the proteins are found in both of them. They ignore design completely. This is used day in and day out in your books. And I have examples of it, but for the sake of time, I will not show it. Just remember this, I love you is formed, the sentence is formed not because of the magnetic process, but the sentence is formed because someone wrote it. The DNA is not functioning like this, not because it's helical, not because of the affinity between the chemicals, but because somebody used this shape to write information of it, and we discovered today this information is 3.2 billion letters long. We discovered also in this information that it is ordered. We discovered also in this information it is needed for every single protein that the body uses. So I just want to leave you with this at the moment. I will just escape and go to the conclusion page of how Zoom share. Take this concept home, please. These are the designs to the theory of evolution, because we have to say it does not account for the origin of life. Design, combinatorics, the origin of information is unexplained, the reduction of a complex system to its components, when they just say that proteins are coming from the same as the ancestor. Natural selection cannot explain the origin of life because it happens after life has started. It's like you're walking and you fall in a ditch, and you say, I'm going to go and bring a ladder from home to come out of the ditch. Well, this is using something you already hasn't solved, which is coming out of the ditch. So natural selection cannot explain the origin of life for that reason, among the others. 6, gaps in the tree of life. 7, there is complex engineering systems in biology, coding, feedback systems, and motor stator systems that require very, very detailed things working together. And I will leave you with this if you have any questions.
Are there any questions for Abuna? If you can come. Abuna, you can see me, right?
I can.
Okay. All right. Someone has a question.
Sure.
I think if I look up here, you can see me head on.
I can.
Hi, Abuna. I have a question. You mentioned an atheist scientist, Francis Collins, I believe his name was.
He's not. He became not an atheist afterwards. Actually, he became Christian. After discovering the DNA sequence, he became Christian.
So how come his theory has not made it through and we still have evolution?
No, he explained the order of the DNA, and that's exactly you're hitting. What's your name?
Oh, Aziz Sterling.
Aziz, you're exactly hitting the nail on the head. Is that the discovery that the DNA is a digital system that is that long, and it has a certain order, is the strongest challenge to evolution, because it brings the question, where did this order come from? And I just explained by the magnetism example here, is that finding this order in the DNA does not explain yet where it came from. It's like when you find a book, but you need to know who wrote it. So we just found the book. What Francis Collins found is the book. I am able to look at the DNA and tell you exactly what is that book, how to be read from the very beginning to the very end. That's what he found. In his own life, he converted to Christianity. And he converted to Christianity actually by reading C.S. Lewis. Now I'm using this, another part related to information theory, but it will be a bit heavy. Even, believe me, this is the lighter part of the talk. Another part which uses scientific methods of information theory to say that this ordered information must be put by an intelligent being because it is ordered. An ordered information or writing a book does not come from an accident. So what scientists is challenged with now, when they discovered the helix, oh, this is beautiful that the information is stored this way. When they discovered the sequence of the information, the one who discovered it wrote a book called The Language of God. But the rest of the field is still denying. Is still denying. But this is exactly what it's about today. Where did the information come from? Who wrote this "I love you" on the fridge? If I have a fridge and a bunch of letters randomly put on the fridge, and somebody came and used these letters and wrote them in a certain order to form a sentence, random processes does not form organized sentence. And if it is, it's an accident. Like if I ask all of you in the church to go out, if the walls of the church are made of paper, and please write randomly whatever comes to mind, just randomly. Maybe I will form the word "of," "the," "may," but I will not form a sentence if each one of you goes randomly write something on the wall. So this is the issue, 3.2 billion letters long, and it carries information. This is really the information enigma. And I showed you here an example of how this information is used just in the case of proteins, and showed you in the case of proteins, the probability of forming a correct protein among other wrong ones challenges the lifetime needed for organisms to exist. So that's the whole essence.
Are there any other questions?
I hope it answered your question. So the person who found it believed, and he believed more because of C.S. Lewis about the right and wrong in absolute sense, but his research helped him to say there is a designer behind this world.
Yes, I had a question about the cosmology argument.
Yeah.
it was like there was a conspiracy similar to this biology textbooks where Einstein created a constant to explain that the universe was not expanding because he knew the implications.
Exactly.
He called that his greatest mistake or greatest lie. He was caught basically in a conspiracy theory with the scientists, and I wondered if the same thing could be true for the biology.
I'll leave you to be the judge. You see, and what's your name?
Ernie Barksdale?
Ernie. So thank you very, very much. I'm actually very encouraged by your response. Yeah, it is the lambda. He called the lambda is my biggest blunder. That's what he called it, because he wanted, just to let you know, the Einstein equation was in parallel developed by a priest, a Belgian priest, and he looked at Einstein's equation and told him, I'm working on the same thing. Your equation describes an expanding universe. And he put a constant to make the equation doesn't describe an expanding universe. And this is actually a tensor equation. I will leave the details to when we talk about cosmology. And later on in his life, he said the lambda was my biggest blunder, because who discovered the expanding universe for sure? Another Russian scientist and the Belgian priest in 1927, and Hubble in the United States when he built the Hubble telescope and he looked at the galaxies. It's a very, very, very interesting history, but that's for cosmology. Any
other questions? Okay, thank you, Abuna.
Thank you all very much. I was always my father.
So now we have lunch downstairs.